Well, thank you everybody for coming. Slowly waking up after arriving earlier this morning. So by question time, hopefully I'll try to make sense. All right, so I'll talk about mainly about a paper we published in 2012, but it's an interesting story. This all started from a previous paper from 2011 that looked at DNA methylation. So I'll give a brief introduction of that paper as well. So the reason we started to look at this idea of antiprofiles and gene expression signatures and so forth is we know that early screening in cancer is arguably perhaps one of the greatest health advances of the last 50 years. However, the things that are in practice in the clinic, which we hope to change, are invasive and expensive, things like mammograms, colonoscopies. And can we find effective, inexpensive, non-invasive screening procedures? That has been perhaps a motivation for a lot of the work that has been done in this community in trying to find gene expression antiprofiles or other genomic measurements that we can use for this type of non-invasive screening. And this is where what we were trying to tackle. And in particular, we were thinking of issues that have arisen in the use of these genomic signatures and understanding that really, except for a very few cases, there hasn't been quite a lot of success in practically using in the clinic gene expression, for example, gene expression signatures for screening or prognosis, et cetera, et cetera. So I'll talk about gene expression antiprofiles and how we think that perhaps rethinking about how we deal with this problem of how to use genomic measurements in prognostic methods might lead us to a better, more successful approach. So as I said, the story begins actually not with a paper that talks about gene expression antiprofiles, but papers that talk about a set of epigenetic experiments, specifically in DNA methylation, that inspired the development of our work in gene expression antiprofiles. Then I'll talk about, that paper was on Nature Genetics 2011. It's a paper we joint work with Casper Hansen, who worked in the lab of our next speaker. So, it's a nice little combination there. And then we'll talk about the gene expression antiprofiles, which is published in 2012. And what we'll talk about is, first, our application to colon cancer screening from peripheral blood measurements, and what we think is some success, but then what we think is really not quite successful. But well, I'll address that in a second. But what I think is actually really quite an interesting result is that perhaps the suggestion that thinking about this antiprofile idea might lead us to kind of universal cancer signatures or set of universal cancer signatures. And more importantly, how the antiprofile approach might lead us to signature robustness and reproducibility, which has been kind of a major problem in the use of genomic signatures in the clinic. So, as I said, the story starts on an earlier paper in 2011, and it's a paper looking at epigenetic gene regulation mechanisms. In particular, we look at DNA methylation, which is a modification directly on DNA molecules, as opposed to other epigenetic type of marks that people look at, like nucleoside positioning or histone modifications. DNA methylation is a modification directly on DNA, which has the peculiarity that we know exactly how it is retained after cell division. So this is a non-genetic. It's not encoded in DNA, but it persists after cell division. So for example, we know it's heavily involved in the ability of cells to retain their state after cell division. So that's how liver cells continue. Well, part of the program that makes liver cells continue to be liver cells after division and brain cells be distinct from liver cells and continue to be brain cells after division. So what did we do in this paper? Well, it was the first whole genome normal tissue to tumor comparison of DNA methylation. It was in colon cancer. And among many of the results that we found, one very, very important result is that although we've known for a long time that in cancer methylation is lost globally, with respect to normal tissues, what we find is that this global hypomethylation is organized into compartments. It's not just a wholesale loss of methylation. So let me illustrate this with this plot. What we have here is a measurement of methylation. Methylation we measure as percentages. We don't measure single cells. We measure a collection of cells. So we're asking what is the percentage of cells in our sample that are methylated at a given position in the genome. So here we would say that at this position in the genome, about 75% of the cells in the 3 normal samples were methylated at that position. Yeah? And then here we would say that at this position, we observed about 50% of the cells in the 3 tumor samples were methylated at that position. So what we observe is this loss of methylation from partial methylation, about 75%, to about 50% in normal, to cancer. It's organized into these compartments. So you see it's a change there, kind of gets back together here, coincides with CPG islands for those of you who know about those. And then there's another compartment here. So it's organized in the genome. All right? So what else did we find in those regions? Well, what we found was an interesting observation. So that if you look at expression in colon within those regions, you observe this interesting pattern that in these regions, most normal samples, these are more than the 3 samples that we were using, which is expression from a large number of samples. So here we're measuring, we're plotting expression for normal versus tumor. In these regions, what we observe is most normal samples are very tightly regulated, almost always silent, but very tightly regulated. Whereas in these regions, for cancer samples, what we observe is that they become activated, but they become activated at seemingly highly variable levels. Right? And in fact, if you look at the genes that show the most hypervariability in colon cancer with respect to colon normal, they are enriched in these compartments. All right? So although we kind of expect that if you look at across different colon tumor samples, you observe high variability, what we're saying is that if you rank genes by their hypervariability, they are in fact enriched in those regions, in these compartments of epigenetic modifications. Right? So there's one more result that we found in that paper. We don't show it here, that we did not whole genome methylation, but targeted methylation studies. But instead of just colon cancer, we also looked at lung, breast, liver, and kidney cancers. And what we find is, again, that in cancer there is hypervariability of methylation, and that they overlap quite highly with the same compartments of hypomethylation in colon cancer. So we think that there's something important going on functionally in these compartments, and that hypervariability is a hallmark. Well, no, I don't want to use that word. That hypervariability is an important characteristic of cancer in specifically those regions. All right? And that is a characteristic across different cancer types. So the same regions show consistent hypervariability across different cancer types. So can we use this insight to define genomic signatures? Well, this is where gene expression antiprofiles come up. What this plot suggests is that there is no, at least if you look at these genes in these regions, there is no colon cancer profile. There is a very clear and distinct normal expression profile. And what distinguishes cancer from that if you look at the aggregate of these genes, it's deviation from that normal profile that defines the cancer phenotype, the colon cancer phenotype. At least it's this set of genes. And these are the set of genes within these regions of hypomethylation in colon. So let's see what we did. Well, let's try to use that idea in classification, in prognosis, etc. So before I go on, an extremely important point. Normalization of gene expression data, in this case from microarray data, is fundamental and key in any of these experiments. All right, we use 3 specific methods developed by our colleague Matthew McCall. The basis of it all is this frozen RMA method, which normalizes microarrays, chips, as a single chip procedure, meaning you give me a single chip and I'll find a way of normalizing it with respect to the data that we used in our experiment. All right, so how did this happen before? People did an experiment, there's a bunch of chips, they normalize together, produce some kind of signature. The next experiment comes in, normalization needs all chips to kind of work together. It doesn't quite work that way in the clinic anymore, right? You can't have all the chips from the thing that you use your signature to derive your signature in the clinic. So you need a way of doing this as a single chip. So if you're interested in doing any kind of clinical usage of expression, highly recommend that you look at these methods. All right, so this is what we used. We have single chip normalization, single chip quality metrics, and a single chip common scale measurement we use called the barcode. So very, very important that you do the proper normalization. This is extremely platform dependent, but this is what we need to do. We need to make sure that we're capturing variability from the specific platform as we need it. Okay, so the Ntype profile call is an extremely simple idea. There's essentially the definition of it, but it's probably easier if I illustrate it. All we do is say we take normal samples, just the normal samples. We define the normal profile. In this case, we do it by just specifying a region of normal expression. We haven't used the colon cancer data at all. And then we say how often does a particular sample, for example, this sample that we've marked here, how often does its expression deviate from the normal region of expression? That's all we're doing. Okay? And then we count the number of genes. And we say if you have a high number of genes, that's indicative of the cancer phenotype. Right? So as you can see, this particular sample is not consistently away from normal in some specific way. It jumps all over the place. What is consistent about it is that more often than not, it will fall out of the normal expression range that we define. And again, point only depends on normal samples. All right, so how good is it? Well, we took 2 colon cancer experiments, completely independent, completely distinct, ran this procedure, classified one completely independent experiment based on the Ntype profile of the other, and obtained these results. We can classify one of the experiments perfectly, the other one fairly well. It says area under the curve is what you're looking for. This is exactly the best you can do. Once you start deviating that way, you start to not classify as well. So again, this is normal versus cancer on tissue. Not particularly the most interesting thing in the world because you can ask a pathologist to do this, but nonetheless, it's a good way of verifying that our methods are doing what we think we are. But most importantly is the stability of this thing. So we looked at the 2 distinct experiments. We defined the normal expression regions for each, and we ask how close are they to each other. The important thing is this only dependent on the normal samples. And we ask, all right, that high range, that limit of the upper range of normal expression, what does it look like for one experiment? And how does it look like for the other completely independent experiment? And we see that for the genes that we use, again, genes within those hypomethylation blocks, they're extremely highly agreeable. Then we looked at peripheral blood samples in colon cancer and applied exactly the same Ntype profile signature that we derived from tissue. So we derived the profile from tissue, applied it to peripheral blood samples, and obtained respectable, not great, but respectable level of prediction. And it's actually just as good as the prediction level obtained by the paper that used exactly these samples to train and test their profile. So it's an indication that it might be a reasonable thing to try. All right, and we didn't engineer this very much. We just took a particular set of these anti-profile genes to report, but had we done a little bit more training, we could have gone a little bit higher in performance. But that's not more important. The really important thing is that the set of genes within those hypomethylation blocks show a distinctly better performance in terms of prediction than if you take signatures of equivalent size from all genes based on hypervariability. So here we choose the most hypervariable of all genes. Here we choose the most hypervariable of just those within hypomethylation blocks, and so forth, so forth, so forth. You see that the prediction from specifically those within the column blocks is much better than those within the set of hypervariable genes across the entire column cancer transcriptome. So, summary. We have antiprofiles that derive accurate and robust signatures of colon cancer. Uses genes within blocks of methylation loss to do this. There is suggestions in peripheral blood samples that this perhaps might have a good application in clinical screening. But the other big result, we saw that in methylation there is a consistency in across cancer types. Can we get universality out of this? So we downloaded about 4,000 arrays, used this single chip normalization procedure, and looked at a large number of normal tissues and a large number of cancer tissues and sought out those genes that consistently, across cancer types, show hypervariability with respect to normal. So what do we see? We see, again, these are normal tissues. White means that the most of the samples for that particular tissue are silenced. Blue means that, dark blue means that almost all the samples for that particular tissue are expressed. So what we see here in normal, mostly silent, genes that are consistently hypervariable are mostly silent in almost all genes, and when expressed, are expressed in a small number of genes. And then what we observe in cancer is that the expression is all over the place, right? So what is this saying? This is saying that genes that are consistently hypervariable across cancer types are tissue specific, right? So how do we determine this? So we did I just counted, what's the ratio of tissue specific genes to non-tissue specific genes when we look at the normal cancer normal variance ratio across all cancer types. So the more consistent hypervariability across cancer types, the higher the probability that a gene is tissue specific. So what are we saying? The genes involved in determining cellular identity are those genes that are hypervariable consistently across different cancer types. All right? So in illustration, this is what this looks like. If we take those genes, it separates normal tissues in cancer. They're hypervariable. There's no specific profile for any of these cancers, well, at least with respect to what the normal profile looks like.
How many genes are you talking about?
In this case, 100 genes, which is the top 100 consistently hypervariable genes. So if we use these genes, can we detect the cancer phenotypes regardless of which organ it is? So we took that experiment. We did a standard 10-fold cross-validation where we take 10% of the data and we ask, can we identify which of these samples are cancer? And we do fairly well, right? So we train on everything else, just on the normal samples of everything else, and then classify. But that's not particularly interesting. What we really wanted to do is, for those experiments for which we had both normal and cancer, the cross-validation experiment that we did is we held out every sample from that tissue, both normal and cancer, trained on every other cancer and its respective normal, and then try to classify. So the adrenal cortex result that we have here of 0.99 AUC, almost perfect classification, was on an antiprofile trained without any adrenal cortex samples at all, either cancer or normal. And for most cancers, except for stomach, which is, and kidney in this case, we did fairly well. So we think that a potential for non-invasive screening through universal signatures might be obtained by concentrating on highly, consistently hypervariable genes, which tend to be tissue-specific genes. So looking forward, we're now thinking about, well, how can we extend this? We have this idea of anomaly classification. What we did here essentially was anomaly detection. We have normals. Can we detect when it deviates enough from normal and call it cancer? The idea here is, can we determine what intermediate or benign lesions look like with respect to actual carcinoma if we measure the deviation from normal? So does deviation from normal correlate with severity in some way? And we're extending support vector machines, which, as if you may know, is based on similarity between samples. And here we have this idea again that the stability that we obtain from all of these experiments is just in the normal sample. So what we do in the anti-profile support vector machine is, when we determine the similarity of two samples, say for example this new sample to this carcinoma or this adenoma, we don't measure similarity directly with these samples. What we do is we measure similarity with respect to the normal samples, stable normal samples, to try to derive a stable classifier for severity based on deviation from normality. So we call that the anti-profile SVM, and the fact that we're using support vector machines, for us machine learning theorists, hacks, kind of gives us a way of talking about robustness and stability, which is really, really nice in this case. These results depended a lot on understanding how a lot of different gene expression experiments look like within specific regions of the genome. We had to develop kind of this idea of being able to visualize how both gene expression and methylation looks like in the genome. So we're developing this visualization tool, which for us, our folks, the more important thing is we hijack tracks on this genome browser and plot our interactive data within those. Makes our life much, much easier. Be happy to talk more about this. It's one of my pet projects, my favorite projects, as we move along. So from this anti-profile work, we're developing this anomaly classification framework, the anti-profile SVM, interactive visualization. There's software involved with everything that I presented. There's the anti-profiles package that implements the classifiers that I've discussed. There are packages in R and bioconductor. There's data that goes along with that, especially that big data set of single chip normalized data. This is really fun to play around with, and our visualization stuff that kind of grew out of this. But for everything else, you can check my webpage, and I'll be happy to take questions now.
Thank you. We have time for some questions. For the classification results, we show cross-validation results. What about the results when applying your models to independent data?
So, a couple of things. All right. When we're developing genomic signatures, right? In this case, this cross-validation idea, when it starts to break down, it's when we use a full data set to do normalization, then hold aside a data set, and then train, and then classify, right? In this case, normalization is done at single chip. Normalization does not depend on what train or test or anything data that you use, right? It's just whatever data was used. I mean, it's a single chip, doesn't depend on training set, right? So when we do this hold a tissue out experiment, we're essentially doing is as if we just downloaded this data from Geo, do this single chip normalization and applied it on the classifier that we learned. So this is as close as independent data that you can get because of this normalization idea. Now, this is all very, very dependent on the platform. So it's a little tricky to say how this would behave, say, if we take some adjuvant arrays and look at it, because this all depends on being able to specify that normal range of expression, and that's very, very platform specific. But there's been a lot of research on this. This is an Affymetrix HGU 133+2. We know exactly how this platform behaves, and this is why.
Is the collection of results when you put all the GDO data sets?
They're not everything in GEO, but it's curated so that we ensure that it's cancer normal and it's high quality and et cetera, et cetera. But yeah. Question?
Yes, question. We know that in cancer, the most interesting thing from the point of view of clinical is to distinguish non-normal from cancer.
Yeah, absolutely.
The different prognosis of the, say, the
Relapse or something like that. Yeah, yeah. So that's what we were doing with this anti-profile SVM. There's results that we have now for relapse time in lung cancer with respect to this universal signature. It looks like this is it's gonna make a difference. I don't think it's as clean as we wish, but there's definitely signal there, and it does show differences between patients that do not relapse, patients that do relapse. Yeah, exactly. Oh, so there is one point that one of the few signatures that has been successful is mammoprint, which is a good prognosis versus bad prognosis. If you read their methods, they are in fact using an anti-profile. The way they do it is that they calculate a profile for normal, for good prognosis, and their score is deviation from that good prognosis. I think that's not an accident. I think that it's not an accident that one of the few successful platforms has been using this idea for a very long time. So that's a very good question. Is the variation that we observe due to the fact that we are observing at the margin a mixture of different subtypes, right? So we looked at, if we have results on this on the paper. So for example, for those samples in GEO for which we have annotation, say, of grade or type or something like that, we did not see a significant, so if we use these hypervariable genes, and then looked at its ability to tell apart type or stage or grade, we did not see it. But we were not, again, we were not looking at the most hypervariable genes. We were looking at the most hypervariable genes within blocks of colon cancer for colon, and those that were tissue specific across different tissues. And consistently, what we think is hypervariability within a single cancer is due to stage, grade, and on these things. But consistent hypervariability, it's a different behavior. Mm-hmm.
And found actually some separation of
Sure. I, again, if we look at the most hypervariable genes, I am not surprised that we will find this, but we were not. We were looking at a very specific set of genes that behave in a particular way, both coming from tissue specificity and both coming from colon cancer hypomethylation.
Yeah, your hypomethylation, is it somehow related to copy number variation?
That result is on the Nature Genetics paper. No. So there is obviously a high variability in copy number in colon cancer, but it's not as well organized into compartments as the hypomethylation blocks were.
So what's the reason for the hypomethylation? Are there specific genes involved that mutate it? Or?
We don't know yet. That's what we're trying to find out. How early it happens, how persistent is it, and all that stuff. New experiments that we're working on right now. But it's a good question. I agree. All right. Okay.